The overall goal of this model is to investigate how basement membrane stiffness and thickness, due to advanced glycation end product exposure, can modulate invasive cell behavior corresponding to the early and advanced stages of metastatic prostate cancer.
This method can help answer key questions in the age-related pathologies field, such as how increased basement membrane stiffness and thickness contributes to cancer progression, and also metabolic disorders like diabetes.
The main advantage of this technique is that it provides a simple method for mimicking the level of stiffness measured in human prostate tumors in the clinical setting.
The implication of this technique extends toward therapy and diagnosis of prostate cancer by improving our understanding about how basement membrane stiffness promotes molecular and cellular changes.
Individuals new to this method may struggle because the model is multidisciplinary, combining techniques from the fields of chemistry, physics, and biology.
We first had the idea for this method when we hypothesized basement membrane stiffness in the prostate gland can promote de novo invasive behavior in non-transformed epithelial cells.
Visual demonstration of this method is critical because interdisciplinary steps need to be described, including cross-linking of basement membrane, followed by 3D cell culture.
Start by preparing an even surface of ice and using it to chill an 8-well chamber glass slide to 4 degrees Celsius. Then, cut off the end of a 200 microliter pipette tip and cool the tip to 4 degrees Celsius.